Hi everyone, my name is Miranda, and the title of my poster is Investigating White Matter Microglia and their Relationship with Beta Amyloid in Alzheimer's Disease. Alzheimer's disease can be characterized by the accumulation of beta amyloid plaques, neurofibrillary tangles, and neuroinflammation, with the most common symptom clinically being the inability to recall new information. White matter inflammation is indicated through white matter inflammatory cells, such as activated microglia. And activated microglia has been identified as being one of the earliest pathological changes to occur in the AD brain. And evidence suggests that white matter activated microglia is associated with impairments in executive function. Specifically, the activated microglia identified in the supraventricular corpus callosum was identified as being the strongest predictor for impairments in executive function. The transgenic APP21 rat model of AD is developed from inbred Fisher 344 rats and overexpresses a mutated form of the human amyloid precursor protein gene. This model, however, does not spontaneously develop amyloid plaque deposition unless a cerebral stressor is introduced, so it is therefore reflective of the earlier stage of disease progression. The double transgenic PS1 rat model also overexpresses a mutated form of the human APP gene in addition to presenilin 1. So in contrast to the APP21 rat model, the double transgenic PS1 does develop amyloid plaque deposition. Previous work in our lab has shown that white matter activated microglia increases in a Fisher 344 wild-type rat model representative of normal aging, and that this age-related increase is further accelerated in a transgenic rat model of AD. Thus, we hypothesized that white matter activated microglia will increase with normal aging and will be further exacerbated in the presence of beta amyloid plaques. To evaluate this hypothesis, we investigated the sex-specific age trajectory of white matter microglia in a Fisher 344 wild-type rat model representative of normal aging and in two rat models of AD, one in an amyloid plaque negative and one in an amyloid plaque positive environment. Rats were aged to 3, 9, and 15 months, and brains were sectioned coronally into 30 micrometer thick sections for immunohistochemistry to be performed. Bregma levels can be identified here, and the anatomical regions that were analyzed included the forceps minor, the cingulum in yellow, the corpus callosum, the supraventricular corpus callosum, the internal capsule, and the hippocampus. Immunohistochemistry was used to detect total microglia, MHC class II activated microglia, and beta amyloid plaques. After immunohistochemistry was performed, images were analyzed using a light microscope, and white matter inflammation analysis was analyzed using Fiji Image J. Anatomical regions of interest were outlined. The background was then subtracted as seen in the image here, and then a threshold with a fixed grayscale predetermined cutoff value was performed to provide us with the percentage area coverage for each anatomical region of interest. Now taking a look at some of our results. So here we can identify our white matter regions of interest for each of our experimental groups. We have our wild types in the first row, the transgenic APP21s in the middle, and the double transgenic PS1 rats in the bottom row. First, having a look at our total white matter microglial analysis. Total microglia was analyzed using a primary antibody against IBA1. And in the corpus callosum, there was a significant decrease in total white matter microglia in the double transgenic PS1 rats when compared to their wild types at 9 months of age. Similarly, when looking at the supraventricular corpus callosum, we saw a significant decrease in total white matter microglia in the transgenic APP21 rats when compared to their wild types at 9 months. At the same time point at 9 months of age, we also saw unique sex-specific differences. So taking a look at activated microglia using a primary antibody against OX6, females displayed a significant increase in activated microglia across several white matter regions of interest when compared to their male counterparts. These white matter regions included the forceps minor, the supraventricular corpus callosum, and the internal capsule. Another interesting observation included the presence of large clusters of activated microglia that were identified across several anatomical regions. So one of our aims was to determine if these regions of activated microglial clustering was associated with regions of beta amyloid plaque deposition in double transgenic PS1 rats at 15 months of age. So taking a look at a section taken from the late hippocampus, interestingly, we can see that there are no amyloid plaques present in the same cortical regions of activated microglial clustering, and this was observed across each anatomical region that was identified. The transgenic APP21 rats displayed increased activated microglia in the internal capsule when compared to their wild types at 9 months of age. However, no other major differences in activated microglia was identified between genotypes. Lastly, having a look at our age-related trajectory of activated microglia. And we saw an age-related increase in activated microglia from 9 to 15 months across each genotype. So we can see our wild types, the transgenic APP21s, and the double transgenic PS1 rats displayed a significant age-related increase in MHC class II activated microglia in several white matter regions, including the corpus callosum and the internal capsule. So our white matter analysis indicates a significant decrease in total microglia in the corpus callosum and supraventricular corpus callosum of transgenic APP21 rats and double transgenic PS1 rats when compared to their wild types at 9 months of age. Female transgenic APP21 rats displayed a significant increase in MHC class II activated microglia in several white matter regions, including the forceps minor, the supraventricular corpus callosum, and the internal capsule when compared to their male counterparts at 9 months of age. Interestingly, regions of activated microglial clustering was not associated with beta amyloid plaque deposition, suggesting additional underlying mechanisms resulting in this microglial clustering. Further histological analysis will include markers of vascular stress. The transgenic APP21 rats displayed a significant increase in activated microglia in the internal capsule when compared to their wild types at 9 months of age. Lastly, wild types, transgenic APP21 and APP-PS1 rats displayed a significant age-related increase in activated microglia from 9 to 15 months of age in white matter regions, including the corpus callosum and the internal capsule. We would like to thank Dr. Lin Wang and Western University's Department of Animal Care and Veterinary Services for their technical support, expertise, and assistance. We would also like to thank our funding sources. I would like to thank you for taking the time to watch my poster presentation, and I hope you have a great day.